Okay, just making sure the PowerPoint's sharing properly. And let's go ahead and continue with this video, or it's a new video. Let's continue talking about white blood cells. I wanted to add something. We were at the very end of a slide and it said, well, some only live for a few days while others live for months or years. Memory B cells would be ones that live a long time. They're going to help, they're going to make plasma cells, antibody factories. These are what remember things you've been exposed to in the past. Anyway, let's move on. All right, so here we have figure 6.6. It's showing the different types of cells, different types of white blood cells, and they're in a different order than I gave you. I gave you never let monkeys eat bananas, the order from most common to least common in a normal body. What you see right here, where is they've broken them down into granulocytes, the ones that are filled with granules, and agranulocytes, the ones that don't have granules. Now, the neutrophil, you're going to recognize from the ridiculously lumpy looking nucleus. The granules, it has are really small and they're difficult to see. But regardless, basophils, eosinophils, neutrophils, these are granulocytes. And the name actually tells you they are cells, site, with granules in them, okay? You could also think of it by remembering fill. A neutrophil is filled with granules, even though they're tiny. An eosinophil is filled with these big red granules. Red because of the eosin dye that we use to stain them with. Basophils are filled with granules. They're filled with purple-looking granules due to the staining process. It's a basic dye, and the name means the lover of that basic chemical. Eosinophils, the name means, oh, the lover of the eosin, the red dye. Anyway, but if you see fill in the end of the name, it is filled with granules. You notice that the agranulocytes, things like lymphocytes, things like monocytes, they don't end in filled because they're not filled with granules. Actually, that's not the real reason why, but there's your mnemonic device to help remember it. Let's move on. All right, so categorization of white blood cells, just like the pretty picture before us said, granulocytes have noticeable granules, and especially the neutrophil lobed nuclei, major lumpy nuclei in neutrophils. Agranulocytes, no granules and no lobes. The nucleus looks like you would expect a nucleus to look like based on all the other things you've seen about cells and their nuclei. All right. So neutrophils are the most common white blood cell. 50 to 70% of the body is normally, or 50 to 70% of your white blood cells are normally neutrophils. They have a really lumpy multi-lobed nucleus. They are your first line shock troops. They are the first thing to go to an infection and try to fight the bacteria. They work through phagocytosis. That phago means to eat, so cyto means cell. This is literally, they work by having the cell eat the invaders. They surround them and try to pull them into the cell and digest them. These things are capable of crawling out of the blood through the capillaries, and so you can also find them in the interstitial fluid and lymph. The interstitial fluid is the fluid around the, sorry, I'm laughing because you might see a dog nose occasionally. The neutrophils will leave the blood and get into the interstitial fluid, the fluid around the cells in the tissue. Let's move on. So there you see one crawling through a gap between two cells in a capillary. There are actually three kinds of capillaries, and this one would be called a continuous because the cells go all the way around. But the white blood cell is squeezing out through that capillary wall and getting into the tissues. All right, eosinophils, they have two big lobes on their nucleus. they have lots of red granules, eosin granules. And if you have an elevated number of eosinophils, your body's saying, oh, there's a parasite. So parasitic infections, parasitic worms, things like that. That's the type of thing they'll fight. They also are involved in allergies. Okay, so basophils, they're the rarest. They're really uncommon. They have a U-shaped nucleus most of the time. They're found in connective tissues, and they release histamine during allergic reactions. That's going to, you're probably familiar with that from hearing about, oh, my nose is runny, I'm taking an antihistamine to stop it. Well, the reason your nose runs is the body's like, I don't know what it is, it's bad, but we got to wash it out. So make lots of mucus and wash out everything in the nose. That's only one of the things it does. Histamine affects the whole body with wash it out. It makes the blood vessels get bigger so you can send more blood. It makes the blood vessels, the walls more porous so you can get the white blood cells out to fight. But it contracts your breathing, your respiratory system, so you're less likely to bring in horrible things. So the whole idea, the histamine released by the basophils is get the bad stuff out, try to stop more bad stuff from coming in. All right, so lymphocytes, the L should remind you that these are little. These are closer to the size of a red blood cell, maybe only a little bit bigger than a red blood cell. These are 25 to 30% of all the white blood cells. And there are two types here, T and B cells. They're named by where they mature. The T cells grow up in the thymus. They leave the bone marrow, go up to the thymus. The B cells mature in the bone. Okay, so B cells are antibody factories. Actually, they are the things that make the plasma cells, or some of them become plasma cells to make antibodies, antibody factory. T cells are, instead of an antibody factory, they are soldiers going out to find and kill things. For instance, there's a type of T cell called a cytotoxic T cell. It will literally go up next to something else that needs to die, and it will spill out poisons to kill it. So, all right, so monocytes are the big ones. Remember I said the M in monocytes should remind you of massive. They're the biggest of the white blood cells, and they grow up to be even bigger than that, to grow up to be a macrophage. The name means the big eater. And those things will eat any invading pathogen, but they'll also eat your old and dead cells. Try to keep things clean. Think of it as a giant Pac-Man. So let's talk about disorders in the body caused by having problems with white blood cells. SCID is severe combined immunodeficiency. That's when you're not making lymphocytes. Leukemia are cancers of the white blood cell. You have white blood cells grow like crazy. You got way more than normal, but they're shaped weird. They don't know how to do their job. And you might get so many of them, they even make it difficult for your blood to have the red blood cells there. So you have anemia and problems like that. The Epstein-Barr virus can help you to develop mono. It affects lymphocytes, and then you get fatigue, sore throat, lymph nodes all swollen, puffy. All right, here's a progress check for y'all. I'm going to be quiet for a few seconds. You can pause it, and then we'll continue. Okay, So now we're going to talk about platelets, which are also called thrombocytes. Those things are not cells. They are pieces of cells. Pieces of cells that they're a little tiny. They're not smooth and round as they move around. They're little fragments. But when you need a blood clot, they get super spiky. They grow big extensions, and those again act like trees across a wound. But what's really weird is they can retract those extensions, so they could go up to a blood clot, grab the different sides of the cell, the two sides of the cells, the cell, then grab the cells on this side and on this side of a cut, and then they can retract and bring the cells closer together. Makes it easier for the clot to heal. Anyway, we'll move on. So we're going to talk about how blood clotting relates to homeostasis. It's pretty simple. Clotting prevents you from bleeding out, running out of blood. That makes it better for things like delivering oxygen. We're going to go through some of the steps in the formation of blood clots and talk about blood clotting disorders. Hemophilia is the big thing. All right. So thrombocytes are pieces of blood cells called megakaryocytes. They're not cells, they're chunks of cells. And the megakaryocytes are not actually supposed to be out of the bone marrow. Anyway, so you make about 2 billion platelets every day, and they are one of the main things in clotting. Prothrombin and fibrinogen are chemicals that also do blood clotting. Pro means before. So prothrombin, is something that will be turned into thrombin during the clotting factor. And I told you before, O-gen is, we're going to generate, we're going to make. So this is another primitive thing that, I should say primitive, early inactive form. Fibrinogen is the inactive form of what will become fibrin, the fibers in the clot. You also need vitamin K for these things to work. All right, so blood clotting is a big deal so that we don't have plasma and blood leaking out of broken blood vessels. There are 13 different clotting factors, calcium, enzymes are all involved in this. It's a chemical cascade. And if you take AP2, they're going to talk about the fast and the slow, the intrinsic and the extrinsic clotting factors and what makes things clot. You get a chemical cascade where a little bit of this causes more of that, causes more of that, causes more of that. In fact, this is one of the very few positive feedback mechanisms in the body. Almost everything is negative. Oh, that's going out of control. Let's negate that by doing this. Blood clotting is so important that a little bit causes more, causes more, causes more. Positive feedback. All right, and again, we already said it requires calcium. So let's go through some simplified steps. You get an injury, that's in 1. 2, platelets, thrombocytes collect there, and it's not showing them, but they get activated, they grow the extensions, platelets and damaged cells release prothrombin activator, which initiates a cascade of chemical reactions. That's the chemical cascade I was talking about. The fibrin threads form and they trap red blood cells behind them. And that's where I kept saying you would get leaves and trash trapped behind the branches across a stream. That's what's happening there. Now, it's not saying it, but when not saying it yet, but when the platelets retract those extensions, it doesn't just bring the sides of the wound together to make it easier to heal. It also squeezes a lot of the fluid out and makes that clot more solid. But we'll get to that. Okay, so blood clotting. Thrombin, so prothrombin is turned into thrombin. which is an enzyme that will turn fibrinogen into fibrin. The fibrin forms the fibers, the branches of the tree across the stream. It provides the framework for the clot. And red blood cells get trapped there. Now, the fibrin is temporary. You've got another enzyme called plasmin that will destroy it. And then you have cells that'll grow. And it says serum escapes from the clot. This is where we talked about, it says, contains all the components of plasma except the fibrinogen and the prothrombin. What I want you to get here is serum escapes from the clot is we are squeezing the fluid out of the clot and making it more solid as the platelets contract, pull their extensions in. All right, let's move on. So there's an electron micrograph. You can actually see all the fibers of the fibrin. That's like a whole bunch of tiny branches and twigs across the stream. And then you see the leaves and trash, the red blood cells getting caught behind that network. All right, so let's talk about blood clotting problems. Thrombocytopenia, the name says you've got too few platelets. Actually, it's too few thrombocytes, which are cell-like. Oh, wow, I didn't mean to expand that. Let's see if I can shrink that back. Yay, okay. This happens because you're not making enough of them. You're not making enough megakaryocytes to break apart into platelets. So it's not happening in the marrow. Or you could have them falling apart prematurely outside the marrow. Either one of those. This could be because you have drugs that make it hard to make megakaryocytes, or it could be because you have something like leukemia where the body is spending all of its time making abnormal white blood cells instead of making the things it should do. What happens in this case when you got too few platelets? You bleed a lot. You have excess bleeding. Thromboembolism is when a thrombus, that's a clot that's in place, breaks off and starts traveling through the body. That's an embolism. And then it plugs the blood vessel. This can cause a stroke. This can cause death. This is bad. All right, hemophilia, genetic disorder that stops you from making clots. The name means the lover of bleeding. So they thought, oh, this person really wants to bleed because they bleed and they bleed and they bleed. It's because they're not able to form clots. All right, check your progress, 6.4. I'm going to pause and give you guys a little bit of time. Hey, we're back. All right. So let's make that more clear. At this point, you should pause the video, and when you're ready to continue, we will continue on. All right, so now we're going to talk about human blood types, and I will let you know that this is a greatly oversimplified thing. What we're going to talk about are the ABO blood types and the RH factor, okay? If you say someone is A positive, that means they make the A antigen and they make the RH. They are positive for the RH, okay? And but we'll get into it. We'll get into it. What I really recommend is watching the Amoeba Sisters video on ABO blood types, and I can throw a link in for you guys to do that. It's really good. Cutesy, cartoony, but solid information. All right. You will predict compatibility of blood types for a given transfusion. I'm going to tell you right now, the reason I said, oh, it's simplified, because we're only talking about these two things, whether someone has ABO or if they're RH. In reality, when we do blood typing, there are, I'm going to go with more than 30 blood typing groups. Actually, I'm going to pause this and check. Okay, I just checked, and Wikipedia is listing 38 different blood typing systems. So yeah, definitely more than 30. And I put a link down below this video. Actually, I put links to the Amoeba Sisters video and the SciShow video on blood typing, and put the link to the 38 different types. But we're going to be focusing on ABO and RH. All right, let's move on. So, okay, so blood types are determined by proteins that are on the surface of the red blood cells. We call those proteins antigens. I want to throw in two terms here for you, antigen and antibody. An antibody is part of your body, and it fights things that might harm your body. It's against those things. But if for mnemonics, just remember an antibody is part of your body. An antigen, or a potential antigen, is something that could generate an immune response, something your body might think doesn't matter, or something your body might think doesn't belong. So these proteins on the surface of red blood cells are potential antigens. They're proteins that might look foreign to your body, might look bad if you received blood. So blood transfusions, giving you blood from another person. This might happen in a hospital when you're sick. And we need to make sure that it's the right type of blood. If your body detects antigens, proteins on the surface of the blood cells that it thinks don't belong, it might attack that blood and clot it up, clump it up into lumps to make it easier for the white blood cells to come and eat it. We call those clumps agglutination, or we call the process of forming those clumps agglutination. And it's bad because you can block capillaries and stuff. Or white blood cells get there and they attack those red blood cells and spill out their hemoglobin and plug up the filtration membrane on your kidneys. It's not good no matter how you go. Anyway, so the ABO blood group is the main one, the famous one. Basically, you have a type A protein that you could have on the surface of the blood cell. You have a type B protein that you could have on the surface of, I said white blood cell, didn't I? You have a type A protein that could be on the surface of a red blood cell. You have a type B protein that could be on the surface of a red blood cell. You might have one, you might have the other. You might have both, and you'd be type AB. And then you might have a situation where you're like, oh, I don't have any. That's type O. Type O doesn't have any of those. Doesn't mean it doesn't have any of the other 38 proteins for the other systems. For instance, there's an O positive. That means you don't have ABO. You know, oh, there's no AB protein. But there is an RH protein. And then there's O negative, which of course doesn't have the ABOs, and it doesn't have the RH. And O negative is what we usually call a universal donor. Although nowadays, now that we know there are so many other systems, we still are going to check compatibility. Anyway, yeah, we don't just say, oh, it's O negative, it's fine. Okay, so blood types that are determined by the presence or absence of the type A protein and the type B protein. If you are type A, that means that your red blood cell has the A antigen on the surface. It's just a glycoprotein, a molecule that has a protein end in the membrane and a sugary end sticking out. And it's the shape of that sugary end that determines what's going on. If it's the type A shape, we say that you have type A blood. If it's the type B shape, then we say you have type B blood. I want you to understand, though, that because you got two sets of DNA, one from daddy and one from mommy, you could have Dad's saying make an A and mom's saying make a B, or vice versa, you know, but one parent says make one and one parent says make the other. You don't make something that's in between then. You do what each parent says. Dad says you need to make A, so you make the A. Mom says you need to make B, so you make the B. So that means that type AB blood, it's not in between. It has A's and it has B's. It doesn't have any molecule that's sort of like an A and a B, and that doesn't happen. All right, so AB blood has the A antigens on the surface of the red blood cells. It has the B antigens on the surface of the red blood cell. And I already told you O is, oh, I don't see any of those antigens. O is, it doesn't make them, doesn't make either one. All right, so here we have type A blood. You have A antigens, and here we have anti-B antibodies. So what we're saying is a person who has type A blood, they are used to having these potential antigens. Doesn't make them sick. But they are looking for, they have hunter-seeker probes, antibodies that are looking for B and will attack it. Actually, that's only, anyway, we'll talk about that later. So type A blood has A antigens and B antibodies. Type B blood has B-shaped antigens on the surface, and it will recognize B as normal if you give them a transfusion of B. But if you give them A, it'll be attacked by the A antibodies, okay? On the next slide is probably going to show you that A and B, if you have AB, you got both. You have the A and you have the B, and you don't have antigens again, so you don't have antibodies against A because A is normal. And you don't have antibodies against B because B is normal. All right. Now, type O, that's O, you don't have any on the surface. Yeah, no antigens. But if you give them type A blood, it'll be attacked because they have anti-A antibodies. If you give them type B blood, that'll be attacked because they have type B antibodies. Antibodies that attack blood with B on the surface. All right. So type A blood makes antibodies against B. In other words, if you have type A blood, you want to receive blood that either has type A on the surface or nothing on the surface. You do not want to receive B. Your body will recognize B as bad, and it will sick the antibodies on the Bs, on the red blood cells with the Bs. If you have type B blood, B is normal. You could receive blood with B, or you could receive blood from O. There's nothing on the surface. That's fine. But if you give that B person type A blood, all the red blood cells are going to get attacked because they've got this A on the surface, and the anti-A antibodies in the receiver's blood, in the B person's blood, will attack the A red blood cells. Now, type AB blood, it doesn't have antibodies against A or B. They're a universal receiver. You could give them A blood and like, fine, I'm used to seeing cells with A. Even B blood, like, no big deal. I'm used to seeing cells with B. You give them O, there's nothing on the surface. That's weird, but they're like, I don't see anything abnormal, because they're like looking for individual proteins, glycoproteins. So it doesn't make any antibodies. Type O blood cannot receive A, cannot receive B, cannot receive AB, because it makes antibodies against A, it makes antibodies against B, and any of those kinds of red blood cells are going to get attacked. So let's move on. So during the transfusion, antibodies on the surface of the, antibodies in the plasma bind to antigens on the surface of the donated cells. If that happens, you've got agglutination, you've got blood clotting going on. So type A blood can't take type B or type AB. Why? Because they both have B, which it attacks. Type B blood cannot receive type A or type AB because type B blood attacks anything with an A. Now type O blood, it's not used to seeing any of those antigens, not A, not B. So O can only receive O. But O can be given to A because there's no B on it to attack. It can be given to B because there's no A to attack. It can be given to AB because AB doesn't attack stuff. So O is a universal donor and AB is a universal receiver. For this one group, the ABO blood group, the RH group, which I'm sure we're about to talk about, it throws a whole monkey wrench into that because you'd be like, oh, well, type AB, it's a universal recipient. They can receive anything. We'll get to it on a slide coming up, but the short version is if you are AB negative, you cannot receive a positive. And it doesn't matter what the positive is. A positive, B positive, O positive, you can't take it. All right. And it says, to be sure, perform a cross match. Mix small bits of blood to test for agglutination. In reality, this is what happens in a hospital. Because you know, unlike what they're saying in the book, they'll just talk about the two groups. You know that there are lots of groups. What they usually do is test for the, say, top 6 groups, most common things to cause problems. And then after all that, they still take a little drop of this blood, a little drop of this blood, mix them together, look at the slides, see if they clot. If they don't clot, you're good. And you can use that blood for that patient who needs it. If they clot, no, find some other blood. Okay, so here we have pretty pictures of the things happening. Type A blood, if it's given more type A, there's nothing for the antibodies to grab. That's cool. Now, type A blood, if it's given to someone who's type B, see how the shape on the end of the antibody is just like it's supposed to grab onto the antigen? Well, it's because it is supposed to. The antibody will grab on the antibody, will come in, grab onto the antigen and hold on to it. And you see how it's a Y-shaped antibody, most are. It's holding on to more than one cell at a time. That's how you form those clots that are so damaging. That's the agglutination. All right, here we talk about a second group. Now remember, there are really 38 groups or more, but the Rh factor is another type of antigen. And if you have it, actually, if you have any of the, like 14, 15, 16, something like that, versions of it, we're going to say you're positive. We just treat it like all the same. If you've got any form of Rh, you are Rh positive. If you don't, you're Rh negative. And Rh only develops after you are exposed to Rh. What that means is those memory B cells we talked about, they remember that. Like, that's not part of my body, and if I ever see it again, we're going to kill it. All right. So what they're talking about here, hemolytic disease of the newborn, you have to understand, if I always say there are 2 people in this world you should never, ever, ever lie to. One is your mechanic, and the other is your doctor. Because they're both going to find out at the end, after doing lots of tests and it costing lots of money, what you were lying about in the first place. You tell the mechanic, oh no, I wasn't doing that with my car. Oh, they'll see the wear and tear, they'll see the damage, they'll know what you were doing with your car. Same thing. If you go to a doctor and the doctor says, have you ever been pregnant before? You answer straight. whether you carry the kid to term or you had a miscarriage, you answer straight. Because if a woman who is Rh negative has a baby that is Rh positive, imagine this is the first baby she's had. Now you got the placenta, and in the placenta, nutrients and oxygen from mommy travel to the baby and go and keep the baby healthy. That's all good. But mommy blood and baby blood don't mix. There's a barrier. Treat my phone here like it's the barrier. There's a barrier between the two bloods. Mommy blood doesn't mix with baby blood. So even if mom is Rh negative, that means she only has the Rh negative version of the gene to give. But she might be married to someone who has an Rh positive. And if that guy's Rh positive, he might pass on the Rh positive version of the gene, cause an Rh positive baby to be inside an Rh negative mama. In that case, if there's no tearing of the placenta, until there's a tearing of the placenta, there's no mixing of the blood. But once we tear the placenta, the blood on this side and the blood on this side, they can mix. This Rh negative person just got exposed to Rh positive blood, and they remember that. Their body remembers. So let's say this is down the road a ways, another few years. Rh negative mom gets pregnant again. But now her body is making antibodies against blood that has Rh positive. If her second child is Rh positive, then the problems start. Because remember, this blood, mommy blood stays here. We have the blood barrier. Daddy blood stays here. Baby blood stays here. There's no mixing of the blood. That's true. But you already know that things can cross this barrier because the oxygen crosses it. The nutrients that the kid needs cross it. The waste from the kid crosses it in the other direction. Stuff can get across, not blood, but antibodies from the blood can. So you really don't want to create a situation where Rh negative mom on second pregnancy with Rh positive child is passing anti-Rh antibodies through the placenta into the baby. Basically, mom's antibodies are attacking and destroying baby blood. And when that happens, there are bad side effects. So let's go ahead and read this. When an Rh negative woman gives birth to an Rh positive fetus, some of the Rh positive blood can leak from the fetus to the mom, causing the mom to make anti-Rh antibodies. When the mother later has a second fetus that is Rh positive, these antibodies can cross the placenta and attack the fetus's red blood cell. The new baby, the second baby, the whole time it's inside mom, it's going to be attacked. Its blood cells will be attacked by antibodies that are crossing the placenta into the baby. This can cause anemia, can cause all kinds of developmental disabilities. It could even kill the child. Fortunately, our technology is advanced enough that if you are honest with the doctor, they can give you a shot of RhoGAM. And if you get a shot of RhoGAM, you're not going to make antibodies for a while, and it will reduce the amount of exposure. Okay, so then the child will be safer, and you'll be safer. Let's move on. All right, okay, they have a pretty picture, but we've already talked about this. Understand that while the blood can't cross the placenta, the antibodies can. The antibodies from the blood can. All right, this is another one of those checks. I want you to go over it, see if you can answer each one of the questions. If not, rewind the video and watch the last section again. I'm going to pause for a little bit and be quiet, and you should pause the video. And when you're ready to continue, you start it up again. Okay, we're back. Let's move on. All right, so homeostasis, that's maintaining. Stasis means we're maintaining things at a constant level, and homeo means self. We're keeping all of the things inside us at a constant level if we can. So we're going to talk here about how the cardiovascular system helps other parts of the body maintain homeostasis. All right, so they have a pretty picture here. You understand that the cardiovascular system, it's helping the digestive system. It's moving things from the digestive system, nutrients from it to all the other systems. You know, the skeletal and the muscular system, they need a lot of calcium that you get from your food. The urinary system, you're taking waste that's produced in the tissues and moving it through the blood to the kidneys to take that stuff out. You also are providing the endocrine system. You're moving things around in the body, moving these chemical signals, these hormones around in the body so that different organ systems can coordinate with each other. All right, let's jump into it. We want you to be able to explain the functions of the cardiovascular system and how they contribute to homeostasis. And this is figure 6.12. Summarize how each system listed in 6.12 interacts with the cardiovascular system. So know that the endocrine system is using the cardiovascular system to transport its signals through the body. Know that the urinary system receives the waste that it needs to take out of the body through the bloodstream, etc. So it wants us to go through them. All right. So understand that the lymphatic system receives excess fluid that is put out from the cardiovascular system. The cardiovascular system, you push fluid out of the capillaries at the beginning of the capillary through hydrostatic pressure, and you bring it back in through osmotic pressure, bring it back from the tissue fluid into the capillaries. But not all of it. You never get back everything that you push out. And so you would have fluid build up in the tissues. To avoid that, we have the lymphatic system, which drains the excess fluid off of the tissues. It takes that extra interstitial fluid and will dump it back. It'll put it through a bunch of lymph nodes to filter it, let the white blood cells look at it, but eventually it dumps it back into the circulatory system. All right, moving on. And that is the end of this chapter. So let's move on, and we can start on chapter 7.